Well, welcome to Chapter 13 on viruses. So if you recall, viruses are non-living things. They're generally composed of a genetic material that is inside of a protein coat. And these viruses then infect pretty much all forms of life. Bacteriophages is the name that we give phages or viruses that infect bacteria. So bacteriophage, phage that infects bacteria. There's other types of viruses that can infect plants, animals, people, everything. One thing that we have talked about before is that we can't simply culture viruses like you can bacteria, because a virus cannot grow and replicate on its own. So that does present some challenges for growing viruses. So generally we do this by either growing bacteria or animal cells in a plate, and then you can grow the viruses in those cells. There's currently about 1,500 known viruses, but they estimate that there's probably 400,000 viruses that exist that we're just not aware of. And we just recently were able to see viruses with the invention of the electron microscopy. And so this is what we're seeing here. This is an actual picture of a virus. So we know that viruses are obligate intracellular parasites. That means that they are required to use a living host cell, and they need to be inside of those cells to replicate. The genetic material inside of viruses can be DNA or RNA. That DNA or RNA can be either single or double-stranded. It can be linear or circular. And this theme is going to continue when we talk about viruses, because there's such a variety in viruses. They do things differently. So we see a large variety here in genetic material. We're going to see a huge variety in the shape of their protein coats. So speaking of protein coats, we call those capsids. So the protein coat that surrounds the genetic material of viruses is called a capsid, and that is made up of capsomeres. And again, there's a huge variety in the shapes of viruses. They have different sizes too. So again, genetic material surrounded by the capsid, and then surrounding that sometimes is an envelope. So some viruses are called enveloped because they have, this is usually made up of the host cell membrane is going to make up the envelope of a virus, but they don't have to. Some species of virus are what we call naked viruses. They don't have an envelope. Just like we talked about with bacteria, viruses have adhesions, and that adhesion and receptor interaction is what's going to determine what host cells that virus can infect. So that means that most viruses are very specific. The specificity can range, so some of them are very specific to a certain type of cell. Some of them have a range of tissues, and some of them can even cross species barriers, as we know that some viruses can infect animals and people. But what determines the specificity of a virus is going to be which receptors it's going to bind to, and what cells have that receptor is going to be within that virus's host range. And we call this tissue tropism. Tissue tropism is the certain cells or tissues that virus can infect. And the host range is going to be determined by those adhesions that we just talked about. So the host attachment sites are going to determine host range, as well as they need to have the right cellular machinery to be able to replicate that virus. So this is a bunch of examples of viruses. I don't need you to memorize or learn anything on this slide. It's just for you to see the variety of viruses here. So this is Bacteriophage T4. You see a lot of Bacteriophage T4. It's very easy to grow in the lab and study in the lab. So we do see this has been highly studied, and you're going to see this as used as an example a lot. So this is what that looks like. And we see it has this head with the tail region, but not all viruses look like that. Some of them have spikes on the outside. Some of them are just straight lines. Some of them are circular. We have this Ebola virus, looks kind of like a worm, but we see that they're all small. So here we have a bacterium for as an example to see that viruses are smaller than bacteria, and of course bacteria are much smaller than a red blood cell. So here, again, I don't need you guys to memorize this. This is just an example of some of the different groups of viruses that we have and the different appearances of all of these viruses, different protein coats and different shapes. We see some of them are a lot more spikier than others. They're, of course, going to have different adhesions to be able to adhere to different cell types, right? Now, I do want you guys to know this. So I want you to just know the 4 morphological classes of viruses. So first we have the helical viruses. These are going to be circular or cylindrical, okay? And polyhedral viruses is the second class. So these have all these different sides to them. And we see these 2 are naked viruses, and this is an enveloped virus. So they can be enveloped or naked. So here we have the polyhedral protein coat here inside, and it's surrounded by the envelope. And here, there's our Bacteriophage T4 again. And these are complex viruses. They have very complex shapes. They're different and unique, so they just throw all of those into the complex virus category. And the 4th one is the enveloped helical viruses. So we're going to see that here, so we can look a little bit closer at it. So this is a helical virus, and it's enveloped. So this is the protein coat here, and we have the envelope on the outside, and this particular virus has spikes, but not all viruses have spikes. So taxonomy of viruses. There's no evidence for a common viral ancestor. So we can't put them in a very easy, simple cladogram showing all the way down to their most common ancestor because there's no evidence for that. So instead of mapping them evolutionarily, we're just going to classify viruses based on their characteristics. So characteristics that we use to classify, we're going to see a nucleic acid, the type of nucleic acid. So whether it's DNA or RNA or what type of RNA that is, that's how we're going to mostly be classifying viruses. You can also classify them based on the strategy of replication as well as morphology. And so we do see families of viruses, and a family of viruses, the name is going to end in viridae. So if you ever see something viridae, so we see down here are a bunch of examples of families of viruses. Pox viridae, herpes viridae, adenoviridae, and papoviridae. And then we also have genus and species, and those will end in virus. So a viral species is a group of viruses that share the same genetic information and ecological host or niche. That's going to be the host, okay? So they're going to share the same host, same tissue, and have very similar genetic information. And so usually when we're talking about a species, you're going to see common names here that you're familiar with. Then we can also go even further, more specific than that, and talk about subspecies. So when we see subspecies, that's going to be designated by a number. So here are a few examples. Herpes viridae, again, is our family. Varicella virus is going to be the species. No, sorry, this is the genus. And then human herpes virus is the species. And then the specific number, HHV1, 2, or 3, would be the subspecies. We see the same thing over here. Retroviridae is the family. Lentivirus, genus. Human immunodeficiency virus is our species, and HIV1 or 2 is the subspecies. So cultivation of viruses, again, they have to be cultivated with their host cells. So for bacteriophages, you can just make a lawn of bacteria, inoculate it with the virus, and then you're going to see the development of plaques. Now in the book, they describe these as patches of dead bacteria. I would describe it in my work with animal viruses more as a clearance. So I don't know if there's a difference between bacteria and animal viruses, or if it's just two different ways of describing it. But with animal viruses, it's more of a clearance of the animal cells surrounding the virus, because it has, in fact, killed all of those cells. So with animal viruses, you're just going to simply grow that tissue on a plate, and then you inoculate it with the virus the exact same way. Or you can also study animal viruses with embryonated eggs or in living animals, assuming that that living animal is within the host range for the virus. So virus identification is not quite as simple as bacterial identification. Bacterial identification, of course, remember we used our different tests, right, to determine what type of bacteria you had, and then you would follow the dichotomous key. We don't do that with viruses. Instead, there's a few different methods to use. One, you can grow that virus on tissue, or you can take a patient tissue sample and look at that tissue and look for cytopathic effects. So here we see where there's multinucleated because those cells have fused. We're going to see that a lot with budding viruses. We haven't talked about that yet, but we will. Here is a patient tissue sample. And so we have a lipid accumulation over here on this side, and that's typical for a hepatitis C infection. So sometimes by just looking at the patient tissue sample and knowing what that virus tissue that's infected with that virus typically looks like, that can also help with identification. There are also a couple of serological tests that you can do. So agglutination tests, of course, you're just going to here take antibodies against that virus, take patient sample, add it, and if the patient sample has antigen for the antibodies that you have, then you will see agglutination. Remember, that's just clumping, so they're all going to clump together. Another test would be ELISA. We have talked about ELISA before. So as a review, remember, typically we have talked about where you're going to add the capture antibody, put in the patient sample, which would have the antigen in it if it is positive for that virus. Then you add the detection antibody, an HRP-linked antibody, which is going to convert substrate if it's present. There's two different ways to do ELISA tests. So the first way is direct ELISA. This is what we have talked about already. Again, antibody you have added, then the patient sample is going to have antigen. That's direct ELISA. Now, indirect ELISA, you, in your test, are going to have antigen for that virus. And then you will take patient sample, blood usually, and what you're testing for in indirect ELISA is you're testing to see whether that patient has antibodies against that virus. So if the patient has antibodies, that means that they have at some point been exposed to that virus. So if you put in your antigen first and then put in the patient sample, and they too, in fact, have antibodies, then you will see the reaction occur, and you will know they have been exposed. Also, most ELISA tests are quantifiable, meaning that it's going to tell you how much antigen that patient has, or antibody if you're doing an indirect ELISA. The way you test that is you put it in a machine and it's going to tell you how strongly it fluoresced. So that's going to tell you how much it has. Lastly, you can identify viruses by PCR. We've talked about PCR. Again, remember, we're amplifying the DNA, the viral DNA, until it reaches a point that is detectable. All right, so viral replications. Again, viruses are obligate intracellular parasites. Viral genome is, so the goal here for the virus is to replicate its genome and to produce more protein codes. So that's how it's going to replicate. Now, its genome is only going to contain a very small amount of genes, much smaller than something that's living, because it doesn't have to code for any of its living processes. All that the virus cares about is replicating. And so those are the only genes that it needs, is genes that are required for replication, and also genes that are required for its protein code. So it's going to have protein, it's going to have genes that encode proteins for the capsid, also proteins that are required for replication, and also it's going to have proteins that are necessary to help it move into and out of the cell. So adhesions, it's going to, of course, encode for adhesions too. When a virus enters into a host cell, there's a couple of things we're going to talk about, these two different methods that it can use. It can either directly use the host cell machinery to replicate itself, or instead of immediately going into a replication phase, some viruses, not all viruses, but some viruses can go dormant, and we're going to talk about that. Again, viruses have to be inside of their host cell in order to replicate or cause any kind of infection outside of the cell. They are inert. And the method of replication is going to depend on the genome. So again, remember we talked about how it can be DNA or RNA, it can be double-stranded or single-stranded, and so this is what this stands for, DS, double-stranded DNA, single-stranded DNA. These are both single-stranded RNA, but that single strand is either positive sense or negative sense. And I don't want to go into details on what that means. You might remember that from previous classes. But positive sense DNA or RNA, well, we can also have positive sense DNA too, but that is going to be the template for translation. And we can also have double-stranded RNA. And based on the type of genetic material that the virus has, that's going to determine what that virus needs to do in order to replicate its genome. So here is a very quick picture showing the difference between those two cycles that we talked about. Viruses can either immediately replicate inside the cell, and that's called lysis, or the lytic cycle. Or a virus can enter into a cell and be dormant, and that's called lysogeny. Now again, remember, not all viruses can do lysogeny, but some of them can. And this is very important when we're talking about transformation. So if you recall, when we were talking about how bacteria share genetic material, we talked about transformation being one of those methods. So here we're only specifically talking about bacteria and bacteriophages. But for bacteria, lysogeny is important for sharing genetic material. Now, the first two steps are always going to be regardless of which direction we're going. The first two steps will always be attachment. This is what we're seeing here in this clip. It's going to attach to the cell using its adhesions, and penetration. So that is how the genetic material will enter into the cell. Now we'll see sometimes what we saw here in this clip is the protein coat will stay outside of the host cell and it will simply inject its genetic material into the cell. That's not always going to happen. Some other viruses, the whole protein coat and everything will enter into the cell. Some viruses will actually be phagocytosed into the cell. They will induce that phagocytosis themselves when they attach to the cell. So different viruses have different methods of entry, but all viruses attach to the cell and get their genetic material inside the cell. It's just that the method will vary sometimes. So here we look at the lytic cycle. The lytic cycle is where we're immediately going to go into the replication phase. So attachment, penetration, and then it jumps right into biosynthesis. So it's going to replicate its genome and synthesize its protein coat. Once we have a lot of replicates of those, then it will mature by assembling new virions. The term virion means a complete virus that's able to exit the cell. So that would include the genome inside of the protein coat. So we're going to assemble new virions, and then eventually that builds up and it will lyse the cell, and of course killing the cell, and then those viruses are released to continue on and infect a new cell. So that's the lytic cycle over here. Going to infect, replicate, lyse, and repeat that. Now, some viruses are able to go into the lysogenic cycle. The lysogenic cycle is where the phage can go into a dormant or latent phase. And to do this, the viral DNA will incorporate itself into the host cell DNA. And there it will stay dormant. And so whenever the host cell replicates, then the viral DNA will also replicate. The host cell will not recognize that there is viral DNA inside of the cell. So it will simply replicate it along with its own DNA. And this can go on for quite some time. And then for sometimes the reason's not known, sometimes we do know, but then the viral DNA will then switch to going into the lytic cycle. And so it will excise the viral DNA out from the host cell DNA, and then it will enter into the lytic phase. And so, for example, I always like to use the cold sore example because most people are familiar. So a cold sore can stay dormant in the lysogenic cycle for some time. And then sometimes it's stress or being out in the sun. But for some reason, then the viral DNA will excise out and enter into the lytic cycle. And then it will replicate. And that's where we will see the actual cold sore develop. So we've talked about lysis, right? That's where the cell membrane will burst and release all of the virions out. But there are some viruses that will exit the cell different, and we call that budding. So instead of just bursting the host cell, they will instead bud out. And we generally will see these in enveloped viruses. And they're going to use the host membrane to form its envelope. And what this does is it allows the host cell to live a little bit longer so that the virus can continue to replicate even longer. As soon as the cell is lysed, it's going to die. So this way it's able to replicate inside the cell and be released without killing the cell immediately. Now, eventually the cell will die because it's using up the cell membrane. And the cell cannot keep replenishing the cell membrane at an equal rate that the viruses are replicating, but it does give it a little bit more time for those viruses to replicate even longer. Okay, here slide we made a couple of changes. So far we've looked at bacteriophages, so we haven't seen a nucleus, okay? So now we're adding in a nucleus. So now you know this is an animal cell, or it could be a plant cell. We can assume that it's an animal just for our class. And we're also going to look at a DNA virus on this slide. So a DNA virus is going to attach and penetrate like normal. And then the genome will actually enter into the nucleus. And in the nucleus, both replication and transcription will occur. So it's going to replicate the viral genome in the nucleus, and it's going to transcribe or produce mRNA. And the mRNA will leave. This is viral mRNA. Will leave and produce capsid proteins, because they're going to encode for the capsid proteins. And so then those capsid proteins will actually enter back into the nucleus. And then they will form virions, and then those will be released. Again, remember, the reason that the mRNA had to leave the nucleus in order for translation to occur is, remember, that's where ribosomes are. And the virus is going to be using the ribosomes. That's what we keep calling host cell machinery. Ribosomes is part of that. It's going to use the host ribosomes in order to make the capsid proteins. And then, of course, those capsid proteins go back into the nucleus for maturation and then release. So that was DNA viruses. Now we have RNA viruses, and we have a few different examples here with a positive sense, single-stranded RNA, negative sense, single-stranded RNA, or double-stranded RNA. Now, there's different things that each type of RNA virus will do, but I just want you to know that what they're trying to do is to make mRNA. And mRNA is going to be like the positive sense strand. And so all three methods are going to work to make that RNA. And then once they make that RNA, and then they will mature and be released like normal, like we've seen. But one thing that's a little bit different here is the RNA viruses have to use RNA-dependent RNA polymerase in order to make RNA from RNA. Normally we see in animal cells, we have DNA makes RNA. Once an RNA is made, you don't replicate RNA. But here in RNA viruses, that's something that needs to happen. So there is no such thing in animals as an RNA-dependent RNA polymerase. So viruses have to bring that with them. They're still going to use the host cell machinery, and they're still going to use the host cell ribosomes, but just in order to replicate the RNA, they need to use RNA-dependent RNA polymerase that they bring with them inside the protein code. There's one more type of a replication for viruses that we're going to talk about, and that is for retroviruses. So retroviruses are single-stranded RNA, and they are going to produce DNA inside of the host cell. In order to do this, they're going to bring along reverse transcriptase. So similar to the RNA-dependent RNA polymerase that we just talked about. But instead of taking RNA and making more RNA, now we're taking RNA and making DNA. And you know that's something that we don't normally do, right, in our host cell, on our host animal cells. You don't take RNA and make DNA. That's backwards. But here, this is what this virus is trying to do. So it needs to bring reverse transcriptase because that's not something you're going to see in a host cell. Reverse transcriptase is going to take the RNA, which is the viral genome, and turn it into DNA. Then that DNA can go into the nucleus. And inside the nucleus, then that's DNA inside the nucleus, and then that can be replicated and transcribed, and now the story is the same that we were talking about with the DNA viruses. And so then that will make mRNAs, that will make capsid proteins, and then you also can make RNA from that DNA and package it all up and release it. Now switching gears here a little bit. There are some viruses that cause cancer in the host cell. And so these are called oncogenic viruses. Onco, you see when we're in reference to cancer. So oncogenic viruses have oncogenes, and oncogenes are genes that cause cancerous-like cells. So they're going to transform the host cell into cancerous cells. And they do that by speeding up replication of the host cell. So it's going to speed up the processes of mitosis. So now these cells have increased growth, loss of contact inhibition. So contact inhibition, in case you don't know, when cells typically grow, once they start touching each other, then that signals to the cell to stop growing. But in transformed cells, that's not going to happen. So then the cells, they're already touching each other, they're confluent, and they should stop growing, but they don't. So they continue to grow, and that's where we get these masses of cells. Transformed cells also have tumor-specific transplant antigens and T antigens. So they're going to have different antigens on the cell surface. And oncogenic viruses are responsible for about 10% of all human cancers. So here are some examples of oncogenic viruses. Papilloma virus causes cervical cancer. Epstein-Barr virus causes Burkitt's lymphoma. Hepatitis B and C cause liver cancer. And human T-cell leukemia virus is also an oncogenic virus. And lastly, we're going to talk about latent versus persistent. So here in red, before we even get to that, we can look at acute infection. So this is something that's going to jump right into the lytic cycle and cause infection immediately, right? The reason we see this drop here in the number of virions is because these virions, they're moving into the cell where they're going to be replicated. So we're measuring virions extracellularly, but possibly in the blood. But then we initially see a drop in the virion levels as they move into the cell's replication. And then as those cells lyse, we're going to see more virions back in the blood. And then, of course, we're going to see a drop as the immune system catches up. Then over here in green, we see latent infection. We've talked about latent infection. So this is where it can lie dormant in the lysogenic cycle. So this is going to stay dormant, and then for some reason, it's going to switch the lytic cycle, cause infection, and then be stopped by the immune system. But then there's also one more type of viral infection. We call these persistent viral infections. So these are not dormant. They are continuously replicating, but they replicate very slowly for a very long time. And they're going to very slowly accumulate. And during this time of replication and accumulation, we might not see any symptoms until eventually they reach a point where they can overcome immunity and then cause disease. So an example of this would be HIV. If you're aware of someone that has been infected with HIV without treatment, it can still take many years for that patient to get sick. And that's because HIV is an example of persistent infection. So it's consistently replicating until it reaches a point where it finally overcomes the immune system, and then we start to see disease. And that is the end of our lecture on viruses. I will see you guys next time.